Theory. The sliding filament theory refers to the mechanism by which muscles are thought to contract to produce force at the cellular level. Let's take a look at the cross-section of a skeletal muscle. The muscle is covered by a protective layer called the epimysium. The epimysium contains many muscle fibers called fasciculi. The fasciculi are covered by another connective tissue called the perimysium. Each bundle of muscle fibers contains between 10 and 100 individual fibers. Each muscle fiber is covered by another connective tissue called the endomysium, which insulates each fiber. The muscle fiber contains hundreds to thousands of myofibrils, and those myofibrils are made of many sarcomeres. The sliding filament theory explains the mechanism of muscle contraction based on muscle proteins that slide past each other to generate movement inside the sarcomere. The sarcomere consists of a few main segments. A band contains the whole thick filament, myosin. I band contains only the thin filament, actin. Z line is the area where two actin filaments connect and transverse the I bands. M line contains the protein called myosin and marks the sarcomere's center. H zone is the area between the M line and the Z line and contains only the myosin. During muscle contraction, the sarcomere is shortened. This happens due to the sliding of the thin filament, actin, over the head of the thick filament, myosin. This is called myosin-actin cycling. How does the pulling-sliding process work? 1. When a nerve signal reaches the muscle cell, calcium is released and causes a change in the tropomyosin molecule, which shifts in position to expose the binding sites of the actin proteins. 2. The myosin heads bind to the binding sites of the actin proteins to form a cross bridge as inorganic phosphate is released. 3. ADP is released, which initiates the power stroke, where the thin filament gets pulled closer toward the midline of a sarcomere. 4. A new ATP molecule binds to the myosin head, causing the separation of the actin-myosin cross bridge. The ATP is subsequently hydrolyzed to ADP and inorganic phosphate, and the energy transferred from the ATP to the myosin head causes it to cock back, like the trigger of a gun cocks the hammer. This contraction cycle will continue until the nerve signal stops.